Let's just dive right into this explainer. Today, we're unpacking something absolutely fascinating, the Emergenet algorithm. If you follow computational virology, you know we've been stuck using subjective heuristics for way too long. Well, Emergenet is fundamentally shifting that paradigm. We are finally moving toward a highly scalable, strictly objective mathematical architecture for predicting the zoonotic spillover of non-human influenza A strains. It's a massive leap forward in how we predict and ultimately prepare for pandemic emergence.
Here's our roadmap for today. We'll methodically cover the theoretical framework of Emergenet first, looking at spillover limits, inference trees, and eDistance before we pivot to its empirical validations and real-world applications.
So starting off, to really appreciate this breakthrough, we first need to confront the severe scalability bottleneck in how we currently evaluate the pandemic potential of animal flu viruses. It is, frankly, a massive hurdle for current public health paradigms.
Influenza A is notoriously tricky, right? With its segmented genome, it's constantly circulating and reassorting out there in animal reservoirs, posing a persistent pandemic threat. So, the million-dollar question is, how do we actually catch the next threat before it jumps? We're literally drowning in next-generation sequencing data from thousands of wild strains every single year. But having the data is one thing. Having the rapid analytical tools to process it without relying on painfully slow, manual in vitro assays is another entirely.
Let's put this into perspective. Right now, the gold standard is the CDC's Influenza Risk Assessment Tool, or IRAT. But IRAT relies heavily on subjective subject matter expert scoring and experimental assays, like ferret transmission studies. Those can take weeks, sometimes months, to compile for just one single strain. Emergenet completely flips this script. It only needs the genomic sequences of the hemagglutinin and neuraminidase surface proteins, and it computes an objective risk score in about 30 seconds per strain. That is a 5 orders of magnitude speedup. You can literally assess a novel strain's pandemic potential in the time it takes to pull an espresso shot. Moving right along, let's peel back the layers and look at the elegant mathematical architecture that actually makes this algorithm tick.
We define Emergenet strictly as an interdependent collection of local predictors. Now, traditional models often assume standard a priori phylogeny or constant mutation rates, but as we know, those often fail spectacularly in real-world virology because of reassortment and selective sweeps. Emergenet instead operates as a recursive forest of conditional inference trees. So in practice, it estimates the conditional residue distribution at each index of a viral sequence by looking at the specific residues residing at other indices. It's brilliant because it captures the complex epistatic constraints dictated by functional viability and host immunity rather than just blindly mapping historical lineage.
Think of these mathematical concepts as the underlying rules of the game for viral evolution. By analyzing point substitutions and indels across the viral sequence, Emergenet maps cyclic dependencies. We're talking about vast epistatic networks here. For example, in an H1N1 hemagglutinin model, the predictor for residue 63 might depend on 155, which in turn depends on 223, which then loops all the way back to 14 and 63. These aren't just random stochastic mutations happening in a vacuum. They're tightly coupled biological dependencies, completely driven by immense structural and immunological selection pressures.
Which brings us to the next phase. Now we transition into the rigorous statistical probabilities that bridge the gap from mathematical sequence dependencies to actual biological replicative fitness. Emergenet introduces a unique adaptive distance metric called eDistance, and this is absolutely crucial. It is fundamentally distinct from a simple Hamming or edit distance that treats all mutations equally. eDistance is calculated as the square root of the Jensen-Shannon divergence of the conditional residue distributions averaged over the whole sequence. Now, why the square root? It's not just a random choice. It's mathematically dictated by Pensker's inequality. Using Pensker's inequality lets us rigorously bound the total variation distance, meaning we can statistically bound the log likelihood of a spontaneous mutational jump from one specific strain to another. To ground this statistical divergence in biology, we define something called persistence probability. This connects the statistical likelihood of a sequence maintaining its exact genetic identity directly to its replicative fitness in a host. If a sequence has a high persistence probability, it's highly fit and well adapted. It basically has zero evolutionary incentive to mutate away from its current state. In essence, it maps the statistical probability directly onto the peaks of the viral fitness landscape. So, when you combine eDistance and persistence probability, we can quantify the exact numerical odds of specific descendants actually arising in the wild.
Theory is great, but it's time for some hard data. Let's see how Emergenet's theoretical framework holds up empirically against decades of historical recommendations. To prove the model works outside of a simulation, researchers applied it to seasonal flu vaccine prediction. Relying solely on sequence data from the prior season, Emergenet tried to forecast the frequency-dominant strains for the upcoming year. And the results? Absolutely striking. Emergenet vastly outpredicts the World Health Organization on seasonal flu. Looking at 2 decades of historical data, Emergenet gave an average match improvement of 3.73 amino acids for Northern Hemisphere H1N1 and 3.32 for H3N2. Think about that. It achieves this strictly computationally, outperforming massive, resource-intensive global surveillance networks using pure mathematics.
Furthermore, the researchers wanted to know exactly which residues were driving these highly accurate predictions, so they utilized a SHAP analysis rooted in cooperative game theory. By treating each amino acid residue as a player contributing to the evolutionary shift, they mathematically proved that the predictive power modulating E-distance is heavily localized within the receptor binding domain, or RBD, of the hemagglutinin protein. This quantitatively confirms what virologists have suspected for a long time. A relatively small subset of surface-exposed residues are responsible for the vast majority of evolutionary phenotypic changes.
With empirical validation thoroughly established on seasonal human flu, it's time to apply this framework to its main objective, computing zoonotic spillover risk from animal reservoirs.
Just look at this staggering number, 6,354. That represents the wild influenza A animal viruses sequenced globally post-2020 that Emergenet successfully analyzed. Because the algorithm only takes mere seconds per strain, it chewed through this massive database with total ease. Doing this with traditional IRAT, in vitro experimental assays, would take decades and millions of dollars in funding.
By evaluating that huge data set, Emergenet assigns an objective, data-driven emergence risk score to every single animal strain, so we can literally pinpoint highly specific high-risk strains. For example, an H7N9 from Hong Kong, an H1N2 from California, and a 2023 H3N2 from swine in Missouri all scored above the critical 7.7 threshold. And here is the kicker. When compared against historical data, these computational scores exhibit a strong Pearson correlation with published, experimentally derived CDC IRAT scores. We are literally achieving laboratory-grade risk assessment purely through scalable mathematics.
To wrap this explainer up, we need to look at how this computational paradigm shift completely redefines our global public health strategy moving forward.
By adopting Emergenet, we can establish a precise, actionable pipeline. First, we continuously and scalably risk rank all newly sequenced wild animal strains in real time. Second, we can deploy targeted inoculations in specific animal hosts, like commercial swine or poultry populations, effectively neutralizing the threat at the reservoir level. For those of us in public health, this means we can design and stockpile preemptive human vaccines for the exact strains most likely to achieve human-to-human transmission long before an outbreak even begins. It's a definitive shift from reactive surveillance to proactive precision mitigation.
Because let's face it, by accurately predicting the evolutionary trajectory of viruses and intervening at the animal reservoir level, we can bypass many of the modern complexities of human vaccine hesitancy entirely. So I'll leave you with this final thought. How might this elegant mathematical architecture ultimately prevent the next pandemic before it ever reaches a human host? The future of virology isn't just happening in the wet lab anymore. It's heavily encoded in the data. Thanks for joining me for this explainer.